what I want to do today is, as usual, I'm going to end up talking about an individual, which is Willhem His. But I want to be talking even more about some of the processes that go on as we sort of refine the technology and refine the approaches to microscopy. I'll start with a couple of thoughts about tissue preparation, because we have a pretty good sense of how to prepare tissues and look at them in sort of contemporary microscopy, even the stuff going back to the fixed slides that we look at, that come out of the 1950s or so, maybe a little bit later than that. They were all prepared by a technique which we basically have standardized over that time. So there's a process, contemporary, I'll call it, the contemporary approach is one fixation, generally these days with an aldehyde fixative. Meaning formaldehyde, glutaraldehyde, things of that sequence. And what those generally are, materials that we know now from lots of work, are involved in cross-linking proteins. And that's been the standard way of preserving tissue. But I want to get back to what people were trying to do beforehand, okay? The next thing that we get involved with is somehow being able to take that tissue and prepare it in a certain way for sectioning. So I'll call it here embedding. Whereas what we could do again for standard light microscope work is embed in paraffin section on a contemporary microtome. And then third process would be staining. And these days, of course, we have not only specific dyes, but we also have a whole array of fluorescent reagents that certainly enrich what we're doing now. Well, if we go back to, even Robert Hooke, so older approaches. And they really do go back as far as Robert Hooke. Alcohol. And lots of efforts to treat with acids, sometimes bases, whatever they could find that would be fairly toxic and destructive in a way, things that would, quote, harden. Because the big concern they had was how to get the tissue soft enough to, hard enough to cut in any way or other. And so you may remember that the remarkable success that Robert Hooke had with looking at the plant tissue, at the cork, was that he figured out a way to cut an extremely thin slice of this bark of cork, and we think of cork now as the sort of stuff that comes out of a wine bottle with a little luck. What he was looking at was a piece of tree bark. And the question is, how do you then proceed to try to analyze that sort of material? For other people who were looking at other kinds of tissues, they had to treat them, they were trying to treat them because they were soft tissues, trying to treat them in something that would make them resistant to being cut so that you could get some sort of cut through them. And so there were a lot of approaches using things like alcohols and acids. And of course, one of the consequences of that was that it caused a precipitation of the cellular content. Rather than preserving what was actually in the cell, what it tended to do was cause it to coagulate. And many people realized this, and there was quite a bit of a question about the best way and how to handle these tissues and how to minimize the amount of coagulation and still get it hard enough to survive. And there are major books that were published in the 19th century about technical approaches, how to do this sort of thing. And that fits in sort of with the embedding and sectioning issue that we still have now, but we seem to have resolved it. That both fixation has been resolved, and we now have a way of embedding tissues that I'll go over perhaps a little bit later as we come to it. Most of the people at this point, in the 1850s, say 1830s to 1890s even, we're trying to figure out, once we get this lump of tissue, how do we support it and how do we get it hard enough to cut? And it is true, people thought about a number of hardening materials. Canada balsam, which is basically the exudate from a tree, was a classic way to try to get things hardened down. And then as it became available, people became aware of paraffin. If I could only spell it correctly. And you'll see references to it as paraffin. Okay. It still doesn't look right. Paraffin. That's better, right? Okay. All right. But their idea, by and large, was that they would use it as a sort of an enveloping material. That is, it would coat the tissue that they cared about, but not necessarily penetrate the tissue. So it formed a sort of again, a rigid support around the tissue. But the tissue inside that support was still subject to whatever had been used to treat it beforehand. So as you remember, an awful lot of what was done was people simply scraped off pieces of tissue and didn't try to cut an entire tissue all the way through. It's only later on, and you'll see how, that people got to being able to make reasonable sections of material. And then in terms of stain, they started with this collection of what you might call natural stains, materials that were extracted from one or another organism. One of the classics that you'll see referred to as cochineal, which is actually the dye that comes off of ladybugs. It's the red dye that's in the shell or the wing cases of ladybugs, among other things. It's an insect product. Obviously, plant products, indigo, and then a few of the chemical things that were identified, like iodine, were used as stains. Okay? Then, towards the end of the 19th century, there was this explosion in dye synthesis. And so artificially constructed chemical dyes were then starting to be applied to tissues and to see whether they brought up individual components. I was able to identify an enormous resource, which I won't share with you because it's all in German, but it lists about 60 different compounds and a little bit of what it is thought that they interact with. As those kinds of processes were going on, techniques were also developing for better ways of cutting tissue. And that's what I wanted to spend some time on, which is talking about the development, a little bit at least, about the microtome. Because if you now think about what we know in terms of biological structure, an enormous amount of it, most of it, as it turns out, certainly tissue structure and eventually cellular structure, are all built on the ability to cut very thin slices of tissue and to cut them reliably. And then you'll see not only to cut them thin, but to cut them in such a way that you can connect one section to the next. You'll see what I mean as we get along. So let me show you a couple of early bits of history. Although this is not the earliest thing I'm going to show you, this is the sample idea, how to cut a piece of tissue. You have a flat plate. You take this flat plate and you drag a knife across it, a sharp edge. And generally, these edges are designed to look sort of like this. Let's see if I can show you the side view of the edge. Because the idea of it is that it tends to be a taper like this and flat on the other side. Okay, so if you look at what's the profile of this knife, you might even be able to make out the idea that it looks something like that. So that you have this flat end that slides across the sample. And okay, not too bad. But supposing you want to be able to actually work your way through a piece of tissue. So the classic early example of a microtome is this one. And I have to show it to you in this sort of pastiche of several different things, just because I couldn't resist. So this was used by a man named John Hill to cut wood for an analysis of, as you see, timber. And he wanted to be able to cut slices that would be extremely thin, but also be, in a sense, something that you could get a stack out of. And so what he did was he had this company, this man by the name of Mr. Cummings, put this thing together, and then a producer named Ramsden actually produced these for sale. But the basic idea was this: there's a cylinder, which is pretty obvious here. What's less obvious is you stick your piece of wood into the cylinder. And while the piece of wood is in the cylinder, you have another plate underneath it here. Here's the screw that you turn to move the piece of wood. And here's the piece of wood itself sticking up. Okay. To move this piece, by turning the screw, the wood goes up. And then this knob across the top is connected to a blade. The blade doesn't show up as well as I would like, but that's what we've got, okay? So there's an edge here, a blade, that you use the handle and you swing it around and you can cut a wedge, a little wedge of wood out. So here's a slice, a small thin piece of wood, and you would see whatever the internal structure was, the veins or the small cellular structures. And then you turn the crank on the bottom and you do it again, and you get another slice out. And that's what Hill used for this actually pretty elaborate book, which all of these things remarkably are things you can download from the web. It's kind of astounding. This particular version It was published in 1770. So even before the optical revolution that we talked about with achromatic and aspheric lenses. So this was a device even then for cutting bits of wood and samples. And it turns out some of these are still preserved. Here's an example of one from a museum in London. And you can see it actually corresponds quite well to this diagram. So this is now considered to be one of the earliest forms of a microtome. Whether it comes in a mahogany box or not is less critical. Okay. Now, in reading over Hill's book, Hill's papers, I was struck by the microscope he used. So I just thought I'd include that in this talk. So this is his microscope. And what he's clear about, I mean, you can try and read this if you want to, but the basic idea is he has a whole bunch of tubes. The tubes here are separable, and you can slip different lenses in at different parts of the tube to get different levels of magnification. And there are a couple of additional lenses drawn down here. His illumination comes from a mirror, as you've seen for a number of others. And he has this big gear system, which actually has two functions. There's a little wheel here, a knob. That knob allows you to sort of ratchet the microscope up and down with your sample over here. Here's your piece of sample. And so the microscope can be ratcheted up and down like this. But he also points out that it's not quite clear in this section, I think, but if you take this screw, this key that's located over here, and loosen it up, this entire toothed wheel can then be rotated so that what you'd end up with is the microscope on its side. So for certain purposes, I'm just going to do it for fun this way. Let's see. There, you rotate it, and now you have the microscope running sideways. If certain samples that you had were better seen sideways, and then you would look in from the side to see it. So I just had to include that because part of our interest is always in these microscopes and the way people look at things. All right, we then come about, this device was actually developed relatively early, but sort of modified and used much later. The description here is that it came about, it's described in this case by a man named Willhem His, and we will talk some more about His in a little bit. But at the moment, I just wanted to get across the idea of how he attempted to get reliable sections that he could then, his hope was to take these sections and then you cut one section like this. Sorry, I'm going to go back to here. You cut one section and then cut another section, hopefully attached to it, and another one all the way along until you had what was then referred to as a ribbon of sections. Early on, this was much harder to do than it is now. But now, traditionally, people pull out a ribbon of samples. On this machine, it seems to me it's very difficult to figure out how to do that. It's also quite a challenge to figure out what, in fact, this machine does. Let me see if I can read. You can try and read this direction, see if you're any better than I am. This text is actually the Google Translate version from a German text from this paper. So I went in and I dug up the paper I've really been trying to make some sense about it. But the basic idea of it is this. This piece, maybe if I lighten the color, it's easier to work with. Okay. This whole piece is what we're seeing on the bottom of the image here. You see there's an S here. That S corresponds to the S down at this end. So we're looking at the same thing, in this case from the side, in this case sort of standing back. And the idea is that this entire apparatus, this entire piece, has on it a sample. That's the sample. And what's not shown in this diagram, but is shown over here, is that there's a screw system. And the screw basically pushes this plate. Terrible design, but that's supposed to look like a screw with a handle on it. You attach it over here. And you turn the handle. And as you turn the handle, the plate moves forward. And as it moves forward, it passes a knife, but you don't see the knife here. What you see here is this interesting arch, right? The sample actually goes through the arch, and on the other side of the arch, here's the arch, right here. In other words, if you were on the other side of this arch, you would see something supporting a knife. And once again, what you do is you, by hand, slam the knife down, using this arch as a guide for how to cut the slice. So it's not a particularly convenient system, because if you want to get continuous slices, you'd have to cut one piece and then move your sample a little bit, cut another piece, move another piece, and get another piece cut. It's not clear what would happen to the tissue. It might stick to the knife blade, but that's not described here. So you end up cutting individual sections and then trying to work with them. So in response to a device like this, which is, from 1870, 1880, at least that's when it was described here. There's also, by the way, some evidence that a similar device was developed by a man named, Czech man named Purkinje, which we know now as Purkinje, who was the anatomist who revealed or discovered the presence of Purkinje cells in the brain, in the cerebellum. And there's some question about who saw this first. Depending on which site you look at, it generally says that His was the inventor of the microtome, but that Purkinje used it more, or not. Depends on the website you go to. These things are remarkably imprecise. At any rate, clearly this was not a fully adequate system. And so, interestingly enough, Minot these days, okay, and Pfeiffer each developed an automated system to do something rather similar. And these were done in the United States. This is all this stuff going on in Europe, and these guys over here, one of them is in Johns Hopkins University down the road in Maryland. The other was at Harvard. Minot was at Harvard. And Pfeiffer, it's sort of interesting in the sense that he was known as a technician. And as a technician, he built this thing, and that was what people ended up with. Let me see if I can give you a better idea of the way these things work, okay? So this is the classic example of what is the Minot version of the rotating microtome. And the idea is you have a crank, and you turn the crank, And in this case, the sample would be sitting on this sort of platform. And as you turn the crank, the sample would go up and down past the knife. So the sample would, you have a sample, it would move down. The knife blade is over here, so the knife would cut it, and then the sample would go down. And then in the design of these things, it would pull it back and then move it forward again. And each time you hit a little, there's a ratchet wheel in the back here. If you hit this ratchet wheel, you'd turn this ratchet wheel a little bit, and that would move the sample forward. So each time you'd crank, you'd move the sample forward. Now, what you would end up with from something like this then was a section on the knife blade. And when you cut the next section, it would push this one out. And so eventually, you'd be creating this kind of ribbon of samples. Okay. So that was basically the idea of certainly the Minot type, but I want to go back for a moment and show you the Pfeiffer one, which is what these things are, although they're much harder to see. It's especially hard to see where the knife is on these. But it turns out, here's a clamp on one of these, see it? And the idea is that you would clamp the knife blade in here. And your sample sits, well, here's the sample, I think. And as you turn the knife, as you turn this knob, this big wheel, the sample would be drawn past the knife. You can see it probably better over here. This little wheel has the sample on it, and here's a sample. And so as this wheel goes around, but the sample doesn't change its position after each cut. So it turns out the rest of this device moves the knife itself. So in one case, you have the knife moving, and in the other case, you have the sample moving. And in this one, sample advance. Now, as it turns out, this design is pretty much what people are using even today. Basically, in sort of unrefined microtomes, that's what these things look like. So we're starting to move, this is interesting, we're starting to move historically towards things that are getting closer to contemporary. We've got a way to go. We've got, another century of the 20th century to get to, but we're moving towards the end of the 19th century. It's kind of an amazing path we're taking in this course. All right, so that brings me to Willhem His. And fascinating man for a number of reasons. Whenever you read any of the biographies of any of these people, they tell you how brilliant they were at birth and how they really picked out their fields when they were very young and they were fluent in many languages. And same sort of story for Willhem His, except he was born not in Germany, but in Basel, Switzerland, and nevertheless moved to Germany, so he moved from Basel. Bern is the other major city in Switzerland, But then he moved to Germany, to Würzburg, and then Berlin, and look at who he ended up working with Johannes Müller and Robert Remak. And of course, Virchow. Okay, and with them all, he took up this major interest in embryogenesis. Okay, so they always make a point that at this young age, he became a professor of anatomy and physiology, now at the University of Basel. And then in the biography that I read, I have to leave some of this interesting terminology. He took up a call from the University of Leipzig, and Leipzig is Germany, to become a professor in Leipzig. This is fairly late in his life. This is in 1872. So he was 45 or something, 46. Well, Leipzig, for people like me at least, Leipzig has a certain resonance because it happens to be the place in which Johann Sebastian Bach had his major achievements. He was the cantor, the chief musician at the Thomaskirche in Leipzig. And so for many people, Leipzig is known just for Bach. But as it turns out, Willhem His was a very important figure there as well. We'll see a little bit more of how these things came together by the end of the lecture. His made some very interesting contributions. Okay? So in addition to the microtome that I showed you just now, he worked with a couple of people to develop what is referred to as a camera lucida. This is one from about 1880 or so. And the idea of a camera lucida is that the image from the microscope, so you have a microscope down here, and you have a sample. The image of that sample is reflected to an eyepiece. Big deal. That's just like any other microscope, right? You look through the eyepiece. But at the same time, there's a device here which is very poorly drawn for some reason, because it doesn't tell you what it really does. Okay? What this thing is here is another prism. So it's a transparent glass prism, right-angled prism. And so the prism is actually constructed roughly in this direction. And the idea of it is that if you have a sample, the image of that sample, favorite optical thing, will go through here to somebody who's looking at it. Somehow that's an eyeball. Well, whatever, okay? So you look at it from the side, but at the same time, this prism reflects also to your eye an image of a piece of paper underneath. Here's the plate with the paper underneath. So the idea is you look through this system and you see over here whatever is on the paper. And what you do is you bring a pencil to the paper and you draw on the paper. You draw on the paper by tracing what you see through the lens. In the absence of a true photographic system. You can manually capture your own selection of outlines of an image. So if you have an image like this, you can draw this outline. Now, the story is then that what His would do is take a series of sections, one after another, like this, and look at them one after another with the camera lucida, and on the same piece of paper, keeping the magnification constant, draw what he saw on the next slice. And continue that with the third slice and the fourth slice. And so what he would end up with is a piece of paper with, first of all, an enlargement of what he was seeing in the microscope. I should point out, of course, these are the lenses that he's using for the microscope. Here's where the sample goes. And so the light goes up here, through here, and then is superimposed. You see both what was in the what comes through the microscope and what's on the paper. And so you can trace it. And so you'd end up with a piece of paper in which you had some sort of, well, to our minds, maybe it looks like a geographic map of some sort, a contour map where you see these various contours showing up. Well, what His was interested in was taking these kinds of images and turning them into a three-dimensional understanding of the tissues. So he would take these sorts of things and either himself or have an artist take these areas. Now, I can't do this with this pen system, but to sort of smudge out these things so that you would actually enhance the three-dimensional aspect of them. And so he would generate some images like the ones on the left. Now, part of what drove His in doing this analysis was a model in his mind that the way the folds in embryogenesis took place had to do with tensions, with pressures and pullings within the sheets of tissue that were there. And so he tried to model it. This is a guy, I am so impressed with his abilities, his thinking about modeling things. So what he would do is, these are the sorts of things, and you'll see more of them in a minute, that had to do with changes in embryonic shape in very early embryos. This is a rubber tube. And he modeled it by taking a rubber tube and pulling on it here to try to get something like these shapes to occur, to try to show that if the appropriate kind of tension was applied, you could start to generate from tubular tissues this kind of structure. That's a very interesting mindset for this man. And so here's an example of what he did, starting with slices of an early embryo. So this, I think you may remember, we had a picture like this last lecture of the very early chick embryo. That's an example of that. And what he did was he took a series of sections through this thing. Remember, we said you're able to cut, because of whatever microtome he was using, he was able to cut a series of sections. Say this one followed by, actually they probably go in this order, here to here, then to here, then to here, and you start to see the folding of the tissue. This is the original notochord, and it goes this way and further and on, until you've cut through this entire structure going this way. He then took another embryo. Clearly couldn't do it once he had cut sections of this one, and cut in the other direction. And so that's what these images are. These are the longitudinal sections of the same thing. And then the question is, how do you put all this together into a model? Well, one way, and many embryologists for many, many years kept the samples on slides just like this, and in their heads kind of put them together so that they would say, oh yeah, this moves this way and I can follow this thing through. His became very interested in creating true three-dimensional models of these things. And so there are quite a few examples now of him creating these structures out of wax. And these are just some examples in which he cut sections like this and assembled them. These are from different ages of embryos. And this is a kind of a masterpiece. What he did is he worked with a sculptor. Once they got these shaded images together, to remember the one I showed you with the tube, this one, is the sort of thing you might do by extension of the shading, by tracing and getting contour lines. But if you work with a talented sculptor, you might be able to convert those shaded images into these three-dimensional ones. And it's an interesting story. This man by the name of Nick Hopwood has been working to sort of revive the idea, the interest in these three-dimensional models. Hopwood is now very interested and has been on a campaign to reactivate the idea of doing these three-dimensional reconstructions. And now, of course, this work was done in 1999. Nowadays, it's relatively trivial to use computer software to create these kinds of three-dimensional images from slices. And people are now doing increasing amounts of that. And if you look at modern developmental biology texts, that's the sort of thing that you start to see. To sort of wrap this thing up, because I couldn't resist, and it turns out to be interestingly true, I've got to throw you this fun fact. Remember that His ended up in Leipzig. When Bach, who is now considered, by lots of people to be the greatest composer who ever lived, at his death, it was just sort of the end of a life. And he had a bunch of sons who were also musicians, but his stuff was thought to be sort of old-fashioned and out of style and not particularly interesting. So his grave was not a big deal. He was buried, but without any kind of special ceremonies, without any kinds of special treatment. And so his grave was apparently lost, as it says in this interesting paper. This is a paper written around the turn of the 20th century by Karl Bayer, full of personal perspective. So he says his Leipziger Landsleute, that means the people who lived with him, didn't really appreciate his greatness, and nobody knew where his grave was. Except there was some way of digging it out of certain records that he was buried not at the St. Thomas Church where he was known to work, but at the St. Johannes Church. And there was, I love this phrase, a poorly founded oral tradition to the effect that he'd been buried 6 steps to the front and from a small entrance on the left side of the church. Well, there's a lot of people buried there. But it then turned out that there was one more bit of rumor about Bach's burial, which is that he had been buried in an oak casket, and not that many were found for that same year. this is a sort of a scary piece of data, right? That there were only 12 out of 1,400 bodies that were found that were buried that year, if I'm reading that correctly, okay? And they found a couple of them, and one of them they opened up, I love this, in the presence of the great anatomist from Leipzig, Professor His. Now, His went on to describe, you know, an elderly gentleman, a skeleton of a certain size, certainly consistent with Bach's age at his death. But he then wanted to figure out, is it really Bach? And he wasn't sure. So what he did, he had this idea. Remember, he's very interested in three-dimensional structure. He said, supposing we have an artist that could take some data that we have and try to construct the face of Sebastian Bach from the skull and see if it looked like him. Wow, okay? So they cheated a little bit because the sculptor, Sefner, was approached and set to work in a room containing some portraits and pictures of Bach. And within a few days, he created a work that looked pretty good, okay? Now, that's from one description, this one from Bayer's text, from Bayer's essay on this. Another essay suggests Hess had actually done a very detailed analysis of the relationship between the skin and the bones of the heads of cadavers. And so what he did was he collected tissue depth data by taking a needle, if you can stand this, I'm sorry, into the tissue of the cadavers and measuring how far in it went. And then they recorded this for a whole bunch of cadavers that they had. And you'll notice it's really sort of sad stuff. These are people who died of suicide, therefore religiously one didn't have to worry about them. And they didn't have to be dealt in a certain way. Boy, we hit interesting issues, social issues. Anyway, the idea is that, and it's not in this particular discussion, it appears that the sculptor was working not only with portraits, but with this data that His had accumulated for some time. So for whatever reason, it was likely to be the genuine article, that it really was Bach in this casket. Interesting question was, how do you do a control? What would you do for a control? And so this is rather sort of odd, but here's the control experiment. Pick someone else whose image is well known and see if you could make the same sort of image from what you think of as a Bach's skull. Okay, and so they tried to make a bust from George Frederick Handel, who was probably much more famous than Bach at the time this work was done. Handel had made an enormous career in England and was quite well known for, we know him now for Messiah, but he had written enormous numbers of operas and other work. He was quite well known as a composer in England. So he was a famous man. And so that was the one that they chose. And I've always been sort of amused by the irony of comparing Bach to Handel in terms of whether their skulls would be similar or not. There was no skull for Handel available, but the question of whether you could reconstruct the face of Handel using the skull of Bach, it's just sort of mind-blowing. At any rate, it's an interesting way to sort of remind you of how the kinds of detailed work that anatomists were doing in the 19th century could really be used for some very interesting purposes. This happens to be a peculiar one, but there we are. So I think what I can do is end at this point. I thought this would be a sort of remarkable way to think about going from the machinery, which was how to cut sections, to eventually getting three-dimensional analysis. Next lecture, I may talk a little bit about some of how this embryogenesis, embryology played out, because it turns out to have been a very interesting theme. It's not directly related to the microscopy theme, but I may introduce it anyway, just to keep things balanced, if you will. The main goal next time will be to start to think about how the famous group of Zeiss and Abbe and Schott really got to finalize, to ultimately design the best, if you will, the ideal microscopes, and what their technology came from and what it was.